In last few lectures, we have started with our discussion on the topic cell division, and under the topic cell division, we have started the discussion about mitosis. So, in yesterday's lecture, we have seen that mitosis is the phase in which the nuclear material will divide and cell starts to multiply. We also got an idea that two types of M phases are there. One is known as mitosis and meiosis. Mitosis is known as equational division and mitosis is also known as somatic cell division. So up till now we have got this particular idea. Along with this, we also got an idea that there is four different phases, particularly four, and we can say five different phases are included in the mitosis. So to complete the mitosis, we can say five different phases are there, and the first one is prophase, second prometaphase, metaphase, anaphase, and the last one is telophase. And after telophase, we know that the cytokinesis will start. So this is nothing but the development, or we can say the progress in mitosis. In last lecture, we discussed in detail how the prophase will take place. Then after prophase, how the prophase leads to prometaphase, and all these things we have discussed. Along with that, we have also discussed that there is a presence of checkpoints at the end of cell, or at the end of that particular phase. So what happens, the checkpoint will check the proper growth of that particular cell, and later on the cell will move for the another phase. So that is nothing but the progression in mitosis. Today we are going to look towards a small video, and after that video we are again going to continue the discussion related to mitosis.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA, chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Hello student, in last lecture we have started. So this is nothing but the process of mitosis which is explained in this particular video, and at the starting of the video we can get idea that how this mitosis will help in the tear and wear process of our body. Suppose we get some injury on our body, that injury will get cured due to the process known as mitosis. So mitosis will help in the increase in number of cell, and that's why we are getting the benefits of that particular mitosis in curing our injuries which is caused by different physical activities. So watch this video 2 times and take down some notes for the further use.